Okay, so we're talking about cells, and there are some things cells do, some activities that we have to talk about that are, it's fairly complex. And so we'll try and go over them real quickly. Remember, cells are the smallest living part of the human body. Everything is made out of cells, or the cells make what is the human body. Even something solid like a bone contains cells, and it is entirely made by cells. Cells have a nucleus, they have cytoplasm surrounding the nucleus, and in that cytoplasm you have organelles, and then they have a plasma membrane. One of the things I've mentioned is mitochondria. Cells have mitochondria, and this is the site of most of the energy production of the cell. Here is really the only part of the cell that needs oxygen, and oxygen is used to break down food molecules, carbohydrates, lipids, even proteins. And what the waste product is carbon dioxide and water. And these in the mitochondria is where the enzymes that make this happen occur. And these enzymes, the thing that they produce that the cell needs is ATP. Essentially, the cell is burning fuel here with oxygen, and the enzymes allow them to capture that energy and make ATP. It's the only place this happens. It provides the vast majority of ATP or energy that the cell needs. Okay, then one of the main activities of all cells is making proteins. And so protein synthesis is a really important thing. This is a two-step process that occurs the same in all cells, all human cells, all other cells. And what happens is genetic information from a DNA strand is copied into a piece of RNA. This is a messenger RNA. And this happens during the first step of the process, which is called transcription. The messenger RNA then leaves the nucleus, okay, and becomes attached to a ribosome in the cytoplasm. And there another type of RNA called a transfer RNA brings amino acids that are specified by the nucleotide sequence on that messenger RNA, and the ribosome links those amino acids together. A protein is just a chain of amino acids, and the exact sequence is determined by a messenger RNA, which is made from a DNA. So that second part of the process is translation. Okay, this is really important. Transcription, it's really a simple process. It's one enzyme, RNA polymerase. And this RNA polymerase causes complementary RNA nucleotides to become associated with the DNA nucleotides. Okay, and so it unwinds the strand, and you get these complementary nucleotides, and this is what forms the messenger RNA. And so that's what happens in transcription. Okay, so a sequence in DNA becomes a sequence in RNA. Okay, in translation, that sequence of RNA nucleotides travels out to the cytoplasm and becomes associated with a ribosome. The ribosome attaches to the messenger RNA, and then the sequence of bases on the messenger RNA has what are called codons, and these are three base sequences that can be read when the messenger RNA is in the ribosome. And transfer RNAs have anticodons that are complementary to that. And there are different tRNAs that bring different amino acids depending on the codons, and those are assembled into a protein. Eventually, there's a sequence on the messenger RNA that means a stop codon is reached, and at that point the ribosome releases, and you get that protein being released, and it can fold up. Okay, so we have how the cells make energy, and now how the cells make proteins. Proteins can be used, they're used for all sorts of jobs. They're used for enzymes, they're used for membrane proteins for transport or for signaling, or structural proteins. So this is a lot of what the cell does. The last thing I want to talk about is how cells reproduce in the cell cycle. Cells go through a cell cycle. Most of the time they're in interphase, which is just a growth phase where a cell has a visible nucleus and is growing. But at some point, the cell grows enough that it decides, I'm going to reproduce. And then it enters an S phase where it copies its DNA. Okay, after it copies the DNA, it enters another growth phase, G2, where it copies the proteins needed to reproduce. And then it goes through mitosis, a splitting of the nuclear material, and then the cytoplasm splits in cytokinesis. So in the S phase, all that happens is the DNA strand unwinds, and again, another enzyme. This time it's called DNA polymerase. This DNA polymerase starts building a new strand next to the old strand by adding complementary nucleotides. And so you get two identical DNA molecules produced from one DNA molecule. Okay, and this is all, you can't really see it because it happens kind of dissolved in the nuclear area of the nucleus. But when the cell is getting ready to split the DNA, the chromosomes become what are called condensed. Normally the chromosomes are unwound so that DNA synthesis can occur. But during mitosis, chromatin starts to wrap itself around proteins and it becomes highly folded and it condenses to form a chromosome. And this is actually two DNA molecules. Each arm of the chromosome is called a sister chromatid. And these are duplicated DNA molecules that are attached at a spot called the centromere. And in mitosis, those condensed chromosomes are lined up at the center of the cell, okay, and then each one splits in half, and so that sister chromatids migrate away from each other. And these migration begins kind of in the center of the cell, and then the two piles of chromosomes migrate to the opposite ends of the cell, and then cytokinesis forms a furrow between them, and the two cells divide. Okay, so I've got all these sequences here. Let's just kind of go through them. The interphase, this is when a cell is growing, and it's the longest part of the cell cycle. The nucleus appears intact. DNA replication may occur, organelles may be duplicated, and the cells likely grow. When a cell has duplicated its DNA and is ready to divide, it then can enter mitosis. And the first stage of mitosis is prophase. And in this phase, the chromosomes become visible and the nuclear membrane disappears. Some microtubules that form centrioles start to migrate apart, and other organelles become disappeared or stored during those times. Okay. Prophase leads to metaphase, where the chromosomes become lined up at the center of the cell and attached to these microtubules that form a spindle between the centrioles. Okay, and then metaphase ends when each chromosome splits at the centromere and sister chromatids or identical chromosomes start moving away from each other. Okay, and this is anaphase. At telophase, they've moved about as far away as they can. Cytokinesis usually begins. There's a cleavage furrow that develops, and a new nuclear membrane starts to form. Okay, and then mitosis is complete. All cell divisions in the body occur this way. We start out as one cell, and through mitosis and differentiation, we become trillions of cells. Okay, so to summarize, cells use oxygen to get energy from food molecules within mitochondria. This produces the ATP cells need. All cells make proteins from information in their DNA, and they do it in a two-step process, transcription and translation. Okay, the enzyme RNA polymerase links RNA nucleotides that are complementary to DNA to make messenger RNA. That's transcription. In the cytoplasm, that messenger RNA attaches to the ribosomes. Then transfer RNAs bring amino acids to be assembled by the ribosomes. That's translation. Okay. In the S phase of interphase, cells use enzymes DNA polymerase to copy DNA by adding complementary nucleotides to an old DNA strand. When cells reproduce, they divide their genetic material equally between two daughter cells by lining up the duplicated chromosomes in the center of the cell. Sister chromatids split apart and migrate to the opposite poles of the cell. Cytokinesis then creates two cells. Okay, we'll stop there. We'll have more in our forum about mitosis and when it goes wrong.